I'm Stephanie Gurr.
And I'm Rachael Bakker. And today we're going to be talking about measuring mRNA distribution in Drosophila imaginal disc tissues. So a protein of interest is called senseless, and it's expressed in two parallel stripes in the Drosophila wing disc. Senseless helps make these cells competent to become neurons, and so it's known as a proneural gene. The expression pattern that you can see here is caused by a signal that diffuses away from the cells in the center line here in between the two stripes. The signal diffuses away, and when it's at appropriate concentrations, it's able to activate senseless protein expression. So we know what the expression pattern of the senseless protein is, but we want to know more about the expression pattern of the mRNA. Particularly, we want to know how the expression pattern of this mRNA might change in response to perturbations of mRNA processing or in the inductive signal that causes senseless to start to be transcribed. So there's some specific things that we'd like to be able to determine. So we want to know whether or not the senseless mRNAs have the same boundaries as the protein expression. We also want to know about how many mRNAs there are per cell, and how this changes as you get away from the center line. We also want to get an idea of what the variability in the number of mRNAs is from cell to cell. So our goal for this project is to just be able to count the mRNAs per cell in this tissue. The way that we're going to go about doing this is using confocal microscopy. So using the microscope, we can take a picture of the tissue and then change the focal plane slightly and take another picture of the tissue. And then if we do this successively, we can get an approximation of the 3D volume of the tissue in X, Y, and in Z. But how are we actually going to count mRNAs? mRNA molecules themselves are actually much smaller than can be identified using a light microscope. So the way that we're going to do this is by using a technique called in situ hybridization. So I bathe the tissue in many small DNA probes that are able to hybridize or bind to the mRNA that I want to pick up. And each small DNA probe is individually labeled with a fluorescent molecule. And when you get enough fluorescent molecules very close together, like bound on an mRNA in this case, they'll appear as a single point spread function of fluorescence under the microscope. And this point spread function will be the size of the smallest object that the microscope can discern or pick up. So you can see what this looks like in this image sac. So we also need to be able to see cells so that we can assign each mRNA spot to a cell. This is not as straightforward as it might be in other contexts because the hybridization procedure to be able to visualize the mRNAs is really hard on the tissue and messes it up. So one way that I've been able to do this acceptably is by using an antibody that can bind to a protein in the nuclear envelope. And in this tissue, the antibody stain looks like it outlines the nuclei and forms these rings. So it's forming these circles, and each circle is a nuclei.
So once we have this imaging data, we write a pipeline in MATLAB to do the image processing. So we find all the mRNA objects from the single molecule FISH data, and we associate them to the respective nuclei. So to do so, we first need to find the centroids of the nuclei. in order to associate the mRNA to these cells. So as you can see from the Z stack, in any given Z stack, we can't see the entire disk. So to get around this, we take the sum project of the stack. This means that we turn the 3D stack into a 2D image where each XY pixel is the sum of the intensities of that XY pixel in every Z stack. So once we do this, we can segment the image in Elastic, and so the image on the right here is the segmentation from Elastic read into MATLAB. So as you can see from the previous slide, the edges of the nuclei are kind of fuzzy, so it's really hard to segment that in MATLAB to find the edges of the nuclei. So to get around this, we instead we use the centroids of each cell to build a Voronoi diagram. A Voronoi diagram is a space-filling lattice where every edge is equidistant between two centroids. So as we said before, we projected the image to an XY plane. So we treat cells as cylinders. Therefore, when we associate mRNA to cells, we use the XY coordinate of the mRNA centroid instead of the XYZ coordinate. So to find mRNA spots, we know that from the image stack that we have that mRNA exists in 3 dimensions. So since our data is good enough to segment this image without having to do any kind of processing, we segment the stack as a 3-dimensional image and then and then find the connected components to define these 3-dimensional objects.
The next step is to try to discern real mRNA spots from spots that we may have segmented that might be like background noise. So to do that, we know that a real mRNA should have a point spread function with a radius of about like 3 to 5 pixels, so about 4 pixels. So we're using that to set an acceptable range for the total number of pixels that a spot must take up to be considered a real mRNA. So we eliminate objects that don't meet this threshold. After doing so, we want to find the average intensity of each of the spots. So this is because the spots themselves are much bigger than an actual mRNA molecule, and so they could theoretically contain more than one molecule. However, if they do, they will contain, on average, twice as many DNA probes and twice as many fluorescent molecules, and so there'll be multiple peaks of average fluorescence within each spot if it contains multiple molecules of mRNA. But if there's a single peak, that indicates that we probably have spots that are single molecules.
So, to calculate the average intensity of each mRNA, we first find the centroid and then build a binary image of the same size as the original stack with a single point at the centroid. Then, using ion dilate in a spherical structural element, we can find every pixel in a sphere of radius 3 away from the centroid of the mRNA. Then we go back to the original image and sum the intensities of every pixel in that spherical structuring element located at the centroid. Using this, we can then find the average intensity of that mRNA find the average intensity of that mRNA. So when we do so, we see that we get a single Gaussian, which would suggest that we only have one mRNA at each spot. But of course, this is all very preliminary. So once we have the mRNA centroids as well as the nuclei as nuclear boundaries as defined by the Voronoi diagram, we can assign mRNAs to their respective cells. So with this data, and with the average intensity at each mRNA, we can begin to address the question of how mRNA expression varies spatially in the imaginal wing disk tissue.